Hello and good afternoon. My name is Mustafa.
I'm Riley.
I'm Jackie.
I'm Arjun. And I'm Majid.
And we'll be discussing filtration of different solute impurities using graphene oxide. So just a quick background about the current state of filtration. Over 780 million people globally go without access to clean water. And because of this, 3.4 million people die every year due to water-related diseases. So there are three common filtration methods used to combat this, which is chlorination, solar water disinfection, and biosand water filters. Chlorination actually has health detriments in the long run, and it does leave an odor and a taste. Solar water disinfection takes 6 hours to just produce one batch of clean water, and biosand water filters have a relatively short lifespan of 1 year, and they're pretty bulky too. So we will be using graphene oxide to construct our filters. So why did we choose it? Graphene oxide has a lot of unique properties. It has a charged oxide layer, which allows it to perform electrostatic interactions and can be used in desalination. And it contains pores on the nanoscale as well. So it has pores that range from 2 to 15 nanometers, which is extremely, extremely tiny. And this proves to be really good for filtration. And it's also really chemically and mechanically stable, so it'll have a longer lifespan as well.
So our research question was, can graphene oxide nanostructures be used to filter out solute impurities in water? And using this research question, we formed a hypothesis, and it was that geo nanofilters will potentially remove impurities with greater efficiency than traditional filtration methods. And so we began by using a nylon filter, and we tried trapping a graphene oxide solution on the filter, so coating it over it, but that didn't work, and the filter got clogged. So we tried to dilute the solution with water, and then we coated it again, but the nylon filter still clogged. And then we also tried to use a sonicator to dilute it even more, but again, it got clogged.
So the next day, we decided to switch up our experimental design by instead just baking another layer on top to see if it would scrape off in a more efficient manner or just at all. But unfortunately, it did not. So then the following day, we decided to use pre-made coffee filters and cut them to be the same size as the filter we were going to be using. And then we put different amounts of drops of our graphene oxide solutions on the filters until we decided that it was saturated enough. And so we put those in the oven, baked off the water, and then we were left with a dried graphene oxide membrane. So this was our experimental setup. As you can see, we had our dried membranes on the left, and then the middle picture is them inside of the filter that we pieced back together with the binder clips, and then we just used the syringe with the solutions as normal.
So the solutions that we used to test the filters were salt water, a bacterial solution, an iron chloride solution, and finally an oil and water mixture.
So going into the experiment, we had expected that we would be able to partially remove the salt ions because we weren't sure how well they would interact electrostatically with our graphene oxide filters. We also thought that we'd be able to clear out the bacteria because we were pretty certain that they'd be big enough to get caught in the filter, and that we could partially filter the iron solution, again unsure about how the ions would interact electrostatically, but we did think we could remove all the oil.
Okay, so for the saltwater solution, we made a really concentrated solution, and we originally planned on using a salinity meter to test the amount of salt in the original solution, and then after we put it through the filter, but we didn't end up getting one of those. So instead, we just reacted it with silver, and as you see here, there's a white precipitate, so there was a reaction, meaning there was still salt in the solution, but we were unable to test the original concentrated solution, so we don't know if there was more salt there and we actually ended up filtering out some of the salt, or that we just didn't remove any of the salt.
So the next solution that we decided to filter was the bacteria, and in this one we just used a visual analysis to see how successful we were And you can see that's the original solution there with some bacteria mixed in, and that's the after we pushed it through our filter. So it's significantly less murky. And just to prove that it wasn't the coffee filter and it was our membrane that was working, we also pushed it through just the coffee filter, and you can see there's very little difference. So this just kind of proves that on a smaller scale, graphene is more reliable than the current uses for filtering bacteria.
Okay, so we also, for our iron solution, we thought that we would be able to filter out some of the iron with our filter, but we were wrong. As you can see over there, the solutions look identical, so we didn't filter out any of the iron. And in the middle picture, we used a zeolite for a different copper lab that we performed last week, and it cleared out the copper, so we know that it is possible to filter it, but the graphene oxide did not work.
And so the last solution that we decided to filter was actually a mixture of canola oil and water. And so what we did was we took off the top of the filter and put drops of oil in, 25 milliliters. And actually only 1 milliliter got through to the final solution. So that means 96% of it was removed. And you can see that's actually more effective than the two current methods. So yeah, it's better for removing oil than predatory microbes.
So, if we could change something with the experiment that we've done, we would try sealing our filter better. Since we did crack open a filter and put in our own, it wasn't sealed around the edges, so the solutions would leak out of the sides. And also, we had no control over the size of the graphene oxide pores, so we didn't know exactly what it would do. And we could also analyze the structure of our graphene oxide membrane to see what exactly the properties it holds.
Okay, just for an overview, we used the graphene oxide filter to try to filter out 4 different solutions because it's typically used just to filter salt out of water. So we wanted to see if the idea of using the membrane could filter out other contaminants. And we really found out that pore size affects the ability of the filter to get rid of contaminants. And we learned that it can filter bacteria. And it has such small pores that we thought the bacteria would get stuck, and we were right. And with the a pure graphene oxide solution, it's possible to remove all ions, but with our diluted solution, some of the ions were still removed. In the future, if someone was planning on building off of our research, or if we could continue to work on this for more time, we probably would use a pure solution just to see that if it made a difference in how well it filtered, and try to use an electron scanning microscope, which you can see on the nanoscale, not the microscale, because since it has nanopores, we wouldn't be able to see all the pores if we just used a regular microscope. We would also try maybe resynthesizing using a different method, so we could maybe adjust the pore size to see how much of a difference it would make in filtering out other contaminants.
So why does this matter? Well, there's actually a lot of real-world applications for filtration, obviously. Some of them are listed here. Iron is a huge contaminator in well water and underprivileged areas. Saltwater deposits can sometimes sneak up and infect fresh water, leading to illness. Oil spills are obviously huge issues with fishing. And the most important one to us was bacteria, since I've personally seen a lot of people get illnesses from contaminated water abroad. So we'd say that was the most important one to filter out. The practice of ion sponging, which is where the electrostatic interactions pull some of the ions out of the solution, is something that's unique to graphene oxide, so that could be the next big step towards clean water. Thank you.
Thank you.
Considering that your main application or one of the main application was bacteria removal, did you study the size of the bacteria or the type of the bacteria that you tested?
So actually, bacteria or all bacteria are around, they're micro-sized, so they're not nano-sized. They're around, they differ in size, but they're all micro-sized. So our filter was more on the nanoscale. And so the pores were too small for the bacteria to go through. So in a sense, yeah, we did study them and we did study their size and we figured out why they didn't pass through.
And if you're looking for the specific details about the bacteria, it was called Lactobacillus rhamnosus, and it was 200 billion CFU, or sorry, million.
So, what was your hypothesis for the iron not not being filtered out? One question, and second was, did you try a concoction, all four of them together, and see what happens? Just curious.
So we didn't actually try a concoction because we wanted to try them separately. But for the reasons why the iron wasn't filtered, we did hypothesize multiple reasons. So if you can go to the slide, the iron, yeah. So we speculated that maybe the pore sizes were too big, and so that allowed the iron ions to actually pass through the pore size. Like if it was too big, it's just going to pass through. We also speculated that maybe there wasn't enough electrostatic interaction between the graphene oxide and the iron itself. And finally, we also speculate that it might have been something to do with our membrane integrity when we synthesized the GO.
So based on what you learned through this experiment, like the gentleman asked, if you mixed everything, what I want to know is there a universal way, like basically, is there one grand filter that can filter everything out?
Oh. No. Since all different types of contaminants have a lot of different traits, you can't just use one filter to filter out all of them. As seen just on this slide, you can see that we couldn't get our membrane to filter out the iron, but a zeolite did work, and activated carbon or activated charcoal is another way to filter out contaminants, but none of them work for all different types because they all have different properties that attract to different contaminants.
Okay, so my second question, real quick, is your iron didn't work. So can you explain why your geo filtered versus something like, I don't know, like a Brita filter that can filter out heavy metals? How does yours, not that it doesn't compare, but why is it that the Brita filter ones can filter out some heavier metals and yours cannot? Like, what's different, chemically speaking?
So the Brita filters are much more like commercialized and like mass produced, so they have a lot of uniformity like across all their filters, which is something that like we couldn't replicate in a lab using like a pipette, basically. Also, I'm unsure about the Brita filter's actual structure and why it's able, like what its pore size is, like and how it stacks up to ours. But I do. Okay, so the Brita filter, I don't know if it has any sort of like electrostatic charge to it or what kind of chemicals they're using to filter it, but ours, because it's graphene oxide, has a very like specific kind of like electrostatic energy that so it can only react and like trap certain things. Yeah.
So the question I have for you guys is, you mentioned the pore size is too big. How would you guys control for pore size?
So using the method that we used, we couldn't like at all. There are different methods out there, like using like different gases that like punch holes through like a certain diameter or whatever. But the way that we did it, there's no way that we could have done it. So I mean,
Yeah, so the way you guys did it, so what would be another way that you guys could do it that would control for pore size?
Well, we originally were going to use the sonicator, which we had, and we listed that it didn't actually end up breaking it up enough, but it does break up the sheet so that it would create different size pores, and depending on how long you would actually use it for would depend on how big the pores got and how broken up the graphene oxide sheet would be.
And another thing that affects the filtration is not just the pore size, but the number of layers, right? So graphene oxide is so thin that the layering is it's staggered so that even if it gets through one layer of the pores, it'll still be caught in the next one because it's staggered like that. But we had such a thin layer and such a diluted concentration that really didn't work as well with this solution.
Thank you.
It's a simple question. On bacteria, you mentioned that you used basically you used what you saw, you used visual distinction, just see before and after. Why didn't you use anything else? Why did you just use visual?
Okay, so we did actually try and use an agar plate to take a couple drops of the bacteria solution like before it's filtered and put it on one side, and then take our filtered bacteria solution that looks much more clear and put it on the other side. But unfortunately, we did not see any results on that agar plate after leaving it for 24, maybe 36 hours. But that could largely be due to the environmental conditions that it was in the lab and improper time given to grow.
All right, nice job. Thank you.